The probiotic that can survive like a tiny biological capsule. What if a probiotic could protect itself from stomach acid by turning into a microscopic survival capsule? That is one reason Bacillus clausii has attracted so much attention. Unlike many familiar probiotics from the Lactobacillus and Bifidobacterium groups, Bacillus clausii is a spore-forming bacterium. In its spore form, it becomes highly resistant to difficult environmental conditions. This may help it survive manufacturing, storage, stomach acid, bile, and parts of the journey through the digestive tract. Bacillus clausii supplements are often promoted for antibiotic-associated diarrhea, digestive discomfort, childhood gastroenteritis, gut microbiome recovery, and immune support. But does the science support all of these claims? The answer is more complicated than a simple yes or no. Research suggests that certain Bacillus clausii preparations may help reduce gastrointestinal side effects during antibiotic treatment. Some studies also report benefits in childhood diarrhea. However, other large clinical trials have found little or no difference compared with placebo. This means Bacillus clausii is not a miracle treatment, and it should not be treated as a replacement for oral rehydration, medical evaluation, or prescribed medication. In this video, we will explore what Bacillus clausii is, how its spores work, what clinical trials say about antibiotics and diarrhea, whether it supports the gut barrier, who should be cautious, and how to choose a better quality probiotic product. Stay until the end because the strain information and safety warnings on the label may be more important than the total number of bacteria inside the bottle. What is Bacillus clausii? Bacillus clausii is a rod-shaped, spore-forming bacterium that has been used in probiotic preparations for several decades. Some newer scientific publications use the name Bacillus clausii because of changes in bacterial classification. However, Bacillus clausii remains the name most consumers, healthcare professionals, and probiotic manufacturers recognize. The bacterium has two main biological forms. The first is the vegetative form. This is the metabolically active form that can grow and interact with its environment. The second is the spore form. A spore is not the same as a fungal spore. It is a highly protected bacterial state designed to survive difficult conditions, i.e., imagine a bacterium placing its genetic material and essential cellular machinery inside a microscopic protective shell. While it is in this state, it is not actively multiplying in the same way as a normal bacterial cell. However, it may become active again when conditions improve. This ability gives Bacillus clausii several practical advantages as a probiotic. The spores may remain stable during storage, tolerate exposure to oxygen, and survive acidic conditions better than many non-spore-forming bacteria. After reaching a more favorable environment in the intestine, some spores may germinate into active bacterial cells. However, this does not necessarily mean that Bacillus clausii permanently colonizes the human digestive tract. Most probiotics should be thought of as temporary visitors rather than permanent residents. They may interact with the existing microbiome, intestinal cells, and immune system while they pass through the digestive tract. Another important point is that Bacillus clausii is not a single, interchangeable ingredient. Clinical evidence is strain-specific and product-specific. Some of the best-known preparations contain a combination of 4 Bacillus clausii strains identified as O/C, N/R, SIN, and T. Other products contain strains such as UBBC-07, 088AE, or different strain combinations. A positive study involving one strain or one 4-strain preparation cannot automatically prove that every product labeled Bacillus clausii will produce the same results. Reviews of the organism repeatedly emphasize differences in strain characteristics, manufacturing quality, antibiotic resistance, and clinical evidence. Why does the spore form matter? To understand the popularity of spore-forming probiotics, we need to consider the journey from the supplement bottle to the intestine. First, the probiotic must remain viable during manufacturing and storage. Next, it must survive exposure to stomach acid. It may then encounter digestive enzymes and bile salts before reaching the intestine. For many microorganisms, this is a stressful journey. Spore-forming Bacillus species have a natural advantage because their protective structure is designed to withstand difficult conditions. This does not mean that every spore in every supplement will successfully germinate. product formulation, storage, strain identity, dose, and manufacturing quality still matter. But spore stability may explain why Bacillus clausii is often available in liquid suspensions, small drinkable vials, capsules, and other shelf-stable formulations. Researchers have also studied Bacillus clausii strains that naturally tolerate several antibiotics. This characteristic may be useful when the probiotic is administered during antibiotic treatment. In theory, a susceptible probiotic could be destroyed by the antibiotic before it has an opportunity to interact with the gut. A carefully characterized antibiotic-tolerant strain may remain viable during treatment. However, the phrase antibiotic-resistant requires caution. Antibiotic resistance is only acceptable in a probiotic when the resistance has been carefully studied and shown to be intrinsic, stable, and unlikely to transfer to harmful bacteria. For example, genomic testing of the Bacillus clausii UBBC-07 strain found resistance to several antibiotics, but the identified resistance characteristics appeared to be chromosomal rather than carried on easily transferable genetic elements. The same investigation did not identify the toxin genes examined by the researchers. These findings supported the safety profile of that specific strain, but they should not be generalized to every untested Bacillus product. The practical lesson is simple. Antibiotic tolerance can be useful, but only when the exact strain has been properly identified and evaluated. How might Bacillus clausii work? Scientists have proposed several possible mechanisms for Bacillus clausii. The first is support for the intestinal barrier. The intestinal lining is not simply a passive tube. It is a highly organized biological barrier made from intestinal cells, protective mucus, and protein structures called tight junctions. Tight junction proteins help control what passes between intestinal cells. When this barrier is disrupted, water loss, inflammation, and increased exposure to microbial products may occur. In a laboratory model of rotavirus infection, a 4-strain Bacillus clausii preparation helped protect intestinal cells from a reduction in barrier integrity. The researchers observed increased expression of mucus-related proteins and tight junction proteins, including occludin and zonula occludins 1. The preparation also reduced some markers of oxidative stress and inflammatory signaling in infected cells. This is biologically interesting, but it is important to understand what an in vitro study means. An in vitro experiment is performed in cells or laboratory systems. It can help scientists identify possible mechanisms, but it cannot prove that the same effect will occur inside every human digestive tract. The second proposed mechanism is stimulation of natural antimicrobial defenses. In the same laboratory research, exposure to Bacillus clausii increased the production of human beta-defensin 2 and LL37. These substances are part of the body's natural defense system and may help intestinal cells respond to microorganisms. The third possible mechanism is competition with less desirable microorganisms. Active Bacillus clausii cells may compete for nutrients or attachment sites. Some strains may also produce substances that inhibit certain microbes. However, these antimicrobial effects are strain-dependent and are not equivalent to taking an antibiotic. The fourth proposed mechanism is immune modulation. Instead of simply boosting the immune system, a better scientific description is that probiotics may influence immune signaling. An excessively weak immune response can be a problem, but an unnecessarily aggressive inflammatory response can also damage tissues. The ideal effect would be better regulation rather than uncontrolled stimulation. The fifth possible mechanism involves microbiome recovery after antibiotics. Antibiotics can reduce bacterial diversity and disturb microbial functions. This disruption is often called antibiotic-associated dysbiosis. A probiotic cannot instantly rebuild the original microbiome. However, certain strains may support the intestinal environment, compete with opportunistic microbes, and reduce symptoms while the ecosystem recovers. These mechanisms provide a reasonable scientific explanation for studying Bacillus clausii, but clinical trials are still required to show whether these laboratory effects produce meaningful benefits for patients. Bacillus clausii and antibiotic-associated diarrhea. Antibiotic-associated diarrhea is one of the most important areas of Bacillus clausii research. Antibiotics are designed to control harmful bacteria, but many antibiotics also affect beneficial or harmless members of the gut microbiome. The resulting disruption can cause loose stools, abdominal discomfort, bloating, nausea, or changes in bowel habits. In some cases, antibiotic-associated diarrhea is mild and temporary. In other cases, severe diarrhea may be associated with pathogens such as Clostridioides difficile and requires medical evaluation. Several Bacillus clausii trials have reported encouraging results. A randomized controlled trial involving adults receiving antibiotics found that the Bacillus clausii 088AE strain reduced the onset and severity of antibiotic-associated diarrhea and related symptoms such as abdominal discomfort, bloating, and flatulence. The probiotic was generally well tolerated in that study. A 2025 systematic review examined the 4-strain O/C, N/R, SIN and T preparation for preventing antibiotic-associated diarrhea and gastrointestinal symptoms. The review reported reductions in diarrhea and symptoms such as nausea, abdominal discomfort, and epigastric pain. However, only 4 sources met the inclusion criteria, including 2 randomized trials, 1 previous meta-analysis, and 1 expert consensus document. That limited evidence base is important. A systematic review can only be as reliable as the studies it includes. The authors concluded that the results were promising, but also called for larger and more diverse randomized trials. Some of the most frequently cited Bacillus clausii studies involve people receiving intensive combination antibiotics for Helicobacter pylori. Helicobacter pylori is a bacterium that can infect the stomach and contribute to gastritis and peptic ulcer disease. Eradication treatment usually requires acid-suppressing medication plus multiple antibiotics. This treatment can be effective, but adverse effects may include diarrhea, nausea, altered taste, and upper abdominal discomfort. In a randomized controlled trial involving 130 patients receiving H. pylori eradication therapy, diarrhea during the first week occurred in approximately 29% of the Bacillus clausii group and 48% of the placebo group. The probiotic group also experienced more days without diarrhea during the first week. An earlier placebo-controlled study also reported fewer gastrointestinal side effects during H. pylori antibiotic therapy when Bacillus clausii was added. These results suggest that specific Bacillus clausii preparations may be helpful for reducing treatment-related side effects. However, reducing side effects is not the same as eradicating Helicobacter pylori. Bacillus clausii should not replace prescribed antibiotics or acid-suppressing treatment. Evidence that it significantly increases H. pylori eradication rates is less certain than evidence concerning symptom reduction. Another common question is whether Bacillus clausii should be taken at the same time as an antibiotic or several hours later. The answer depends on the exact strain, the antibiotic, and the product instructions. Some Bacillus clausii strains were selected partly because of their antibiotic tolerance, but this does not mean they are resistant to every medication. Patients should follow the product label and advice from their doctor or pharmacist rather than assuming that all probiotic strains can be taken in the same way. Does Bacillus clausii treat acute diarrhea in children? This is where the evidence becomes more complicated. A 2018 systematic review and meta-analysis examined randomized trials of Bacillus clausii for acute childhood diarrhea. The review found a possible reduction in diarrhea duration and stool frequency, but the size of the effect was generally modest, and the authors noted limitations in the available trials. A trial of the UBBC-07 strain also reported reductions in diarrhea severity among children receiving the probiotic. More recently, a 2025 preparation-specific review and meta-analysis reported improvements in diarrhea duration, number of stools, and hospital stay in children receiving a particular 4-strain Bacillus clausii preparation. The authors concluded that the analyzed preparation appeared effective and well-tolerated, while also stating that additional high-quality randomized trials and direct comparisons with other probiotics were needed. However, not every major trial has been positive. A large Phase 3 randomized double-blind trial published in 2022 included more than 450 children with acute diarrhea. Approximately 97% of children in the Bacillus clausii group and 98% in the placebo group recovered during the 120-hour study period. Median recovery time was approximately 42.8 hours with Bacillus clausii and 42.1 hours with placebo. The difference was not clinically or statistically meaningful. Another randomized comparison found that Saccharomyces boulardii CN-CMI 745 performed better than a Bacillus clausii mixture for reducing the duration of pediatric acute diarrhea. How can some studies find a benefit while others do not? There are several possible reasons. Different studies use different strains, doses, formulations, and treatment durations. Children may have diarrhea from different causes. Viral gastroenteritis, bacterial infections, antibiotic-associated diarrhea, food intolerance, and chronic gastrointestinal disorders are not identical conditions. The age of the child, nutritional status, baseline microbiome, timing of treatment, and use of oral rehydration can also influence results. Study quality matters as well. Small or unblinded studies may report larger effects than carefully controlled, adequately powered trials. The most responsible conclusion is that Bacillus clausii may provide a modest benefit in some forms of childhood diarrhea, but the results are inconsistent and product-specific. Most importantly, a probiotic must never delay oral rehydration or medical care. For children with gastroenteritis, oral rehydration solution remains the foundation of treatment. CDC guidance emphasizes oral rehydration and continued nutritional support, while current NICE primary care guidance in the United Kingdom does not routinely recommend probiotics for childhood gastroenteritis. Urgent medical help may be needed if a child becomes unusually sleepy, cannot keep fluids down, produces very little urine, develops blood in the stool, has severe abdominal pain, or shows signs of dehydration. What about irritable bowel syndrome? Bacillus clausii is sometimes marketed for bloating, abdominal pain, and irritable bowel syndrome, commonly known as IBS. However, evidence for IBS is less convincing than evidence for antibiotic-related digestive symptoms. A Phase 3, randomized, double-blind trial examined a 4-strain Bacillus clausii preparation in children and adolescents with IBS. All participants also received conventional care. After 8 weeks, approximately 73.6% of the Bacillus clausii group and 78.5% of the placebo group reported improvement. There were no significant differences between groups in overall symptom improvement, stool characteristics, bloating, abdominal pain, or IBS-related behavior. This does not prove that Bacillus clausii can never help an individual with IBS. IBS is a complex condition with several subtypes and many possible triggers. But the study does mean that Bacillus clausii should not be promoted as a proven general treatment for pediatric IBS. Claims involving weight loss, anxiety, depression, allergy prevention, skin health, or major immune enhancement are even less established. Laboratory mechanisms and microbiome theories can generate research questions, but they are not substitutes for well-designed human trials. Is Bacillus clausii safe? In clinical trials involving generally healthy adults and children, Bacillus clausii has usually been well tolerated. Reported side effects are often mild and may include gas, bloating, or temporary digestive discomfort. But generally safe does not mean risk-free for everyone. Rare cases of Bacillus clausii bacteremia and sepsis have been reported. Bacteremia means that bacteria are detected in the bloodstream. Sepsis is a dangerous systemic response to infection. Published cases have involved infants, critically ill patients, people with serious underlying conditions, and some individuals with disrupted gastrointestinal barriers. Cases have also been reported in intensive care settings after probiotic administration. A case has even been described in a person who was not considered immunocompromised but had active gastrointestinal disease, demonstrating that risk cannot be predicted perfectly. These events appear rare compared with the widespread use of probiotics, but they should not be ignored. Medical advice is especially important before using a live probiotic in premature infants, critically ill or hospitalized patients, people with severely weakened immune systems, patients with central venous catheters, people with severe intestinal inflammation or major damage to the gut barrier, and patients recovering from major surgery. A high-quality probiotic should also be free from contamination and accurately labeled. Do not use a leaking, swollen, damaged, or expired liquid vial. Follow the stated storage instructions. Do not mix the product into a very hot drink unless the manufacturer specifically says this is acceptable because excessive heat may affect viability. Pregnant or breastfeeding individuals, parents giving a probiotic to a young infant, and anyone taking multiple medications should discuss the product with a qualified healthcare professional. How to choose a Bacillus clausii product. When comparing probiotic products, many consumers focus only on the largest CFU number. CFU means colony-forming units. It is an estimate of the number of viable microorganisms capable of growing under specific laboratory conditions. A higher CFU count is not automatically better. The first thing to check is the full strain information. Look for more than the words Bacillus clausii. A better label identifies the strain or strain combination such as O/C/N/R SINNT UBBC-07 or another documented strain. Next, check whether human studies have tested that exact strain for the condition you are interested in. A strain studied for antibiotic-associated diarrhea is not automatically proven for IBS, constipation, or immune support. Look for a viable count that is guaranteed through the end of shelf life, rather than only at the time of manufacture. Check the expiration date and storage instructions. Look for clear manufacturer contact information and quality control standards. Avoid products making unrealistic promises such as permanently repairs the microbiome, works for every type of diarrhea, eliminates H. pylori naturally, or replaces antibiotics. For antibiotic-associated digestive symptoms, discuss the timing of the probiotic with a pharmacist or clinician. The correct schedule may depend on both the probiotic strain and the antibiotic. For acute diarrhea, hydration comes first. For persistent diarrhea, blood in the stool, high fever, severe pain, repeated vomiting, or symptoms of dehydration, seek medical advice rather than relying on a supplement. And remember that a probiotic is only one small part of gut health. A diverse diet containing appropriate fiber, adequate hydration, regular movement, sufficient sleep, and responsible antibiotic use usually matters more than continuously switching from one probiotic supplement to another. Common mistakes people make with Bacillus clausii. The first mistake is assuming that every Bacillus clausii supplement is identical. Different products may contain different strains, doses, and manufacturing standards. The second mistake is looking only at the total CFU count. A clinically studied strain at an appropriate dose may be more meaningful than an extremely high number of poorly identified organisms. The third mistake is using a probiotic instead of oral rehydration during diarrhea. A probiotic may sometimes be used as an adjunct, but it does not replace the water and electrolytes lost through diarrhea and vomiting. The fourth mistake is assuming that natural means completely safe. Bacillus clausii is a living microorganism. Although serious complications are uncommon, invasive infections have been reported. The fifth mistake is using evidence from one condition to promote another. Evidence concerning antibiotic-associated diarrhea does not prove benefits for weight loss, mood, skin health, or every form of IBS. The sixth mistake is expecting instant microbiome restoration. The gut microbiome is an ecosystem containing hundreds of interacting species. No single probiotic can rebuild the entire community overnight. The final mistake is ignoring the expiration date and storage instructions. A probiotic only has a reasonable chance of working when the stated microorganisms remain viable, correctly identified, and uncontaminated. Final evidence-based summary. So, is Bacillus clausii a good probiotic? For certain people and certain purposes, it may be useful. The most encouraging human evidence relates to reducing diarrhea and gastrointestinal side effects during antibiotic treatment, including intensive Helicobacter pylori eradication therapy. Evidence for acute childhood diarrhea is mixed. Some trials and meta-analyses report modest improvements, while a large placebo-controlled study found almost identical recovery times. Evidence for IBS and many broader health claims remains limited or negative. Its spore-forming biology offers practical advantages, including stability and survival through difficult gastrointestinal conditions. Laboratory studies also suggest possible effects on the intestinal barrier, mucus production, tight junction proteins, antimicrobial defenses, and inflammatory signaling. But laboratory mechanisms do not guarantee a meaningful clinical result. The exact strain, formulation, dose, condition, and patient all matter. For healthy individuals, Bacillus clausii is generally well tolerated. However, people who are critically ill, severely immunocompromised, or medically vulnerable should only use live probiotics under professional supervision. The best question is not simply, does Bacillus clausii work? The better question is, which Bacillus clausii strain was studied, for which condition, at what dose, in which type of patient, and compared with what treatment? That is how evidence-based probiotic decisions should be made. This video is for education only and is not a substitute for diagnosis or personalized medical advice. If you want to understand how your body really works and the benefits of probiotics, subscribe to this channel because we turn complex scientific topics into simple and powerful information that you can actually use. And if you found this helpful, share it with someone who cares about their health.